Under normal circumstances, the tissues of the body are kept under surveillance by antigen-specific memory T cells that migrate out of blood vessels into the extracellular space. If they do not encounter the antigen that they are programmed to recognize, they return to the circulation via the lymphatic system. When foreign antigen enters the body, it is taken up by antigen-presenting cells, processed, and presented at the cell surface as MHC peptide complexes. When memory T cells specific for this antigen encounter the processed antigen, they activate the production of a number of cytokines, including TNF-alpha and IL-1, which act on local blood vessels. TNF-alpha and IL-1 activate the vascular endothelium, which results in a number of inflammatory effects. The tight junctions between the endothelial cells loosen, allowing fluid to pass out of the blood vessels and cause local swelling. Leakage of red blood cells causes the local tissue to become reddened in appearance. The activated endothelium also expresses adhesion molecules that allow circulating phagocytes to migrate out of the blood vessel at the inflammatory site. This process takes between 1 and 3 days, which is why it is called the delayed hypersensitivity response. The extravasated cells phagocytose antigen, and in the case of macrophages, can act as additional antigen-presenting cells. Memory and effector T cells also recognize the adhesion molecules expressed by the vascular endothelium and migrate into the tissue, continuing the process of inflammation.